After studying this particular module, you shall be able to know the 4 levels of protein structure, learn about primary structure of a protein, learn about the structures of hair and silk fibrion, learn about the quaternary structure of protein taking myoglobin and hemoglobin examples. Protein function is highly governed by its stable structure. This structure has basically 4 levels. First is the primary, second is the secondary, third is the tertiary, and fourth is the quaternary levels of protein structure. Protein structure is stabilized by multiple weak interactions. Primary structure is basically the sequence of amino acids joined to one another by peptide bonds. Secondary structure, on the other hand, is the local folding of a part of a polypeptide. Next level, which is the tertiary level, it is basically the mixture of alpha helix and beta sheet, which we are going to learn in this particular module, while quaternary structure is the subunit composition of a protein. In this module, we will move deeper into the deeper insights of these 4 levels of protein structure. So, coming on to the first level of protein structure, it is called the primary structure of proteins. Primary structure is basically gives you a descriptive account of sequence of amino acids and all the covalent bonds, let it be peptide bonds or the disulfide bonds linking the various amino acids in the polypeptide chain. Secondary structure, which is yet another level. Structural patterns is basically the structural patterns made from the arrangement of amino acid residues. The next level, which is the tertiary structure, it is the 3-dimensional folding of a protein. Next level and the last level, which is the quaternary structure. So, this takes into account the spatial arrangement of various subunits. Let us take one example to understand what is this primary, secondary, tertiary and quaternary structures. Hemoglobin, as we know, which is the oxygen carrier which is present in our blood in RBCs, it is basically composed of 2 subunits. First is the alpha and the other one is the beta subunit. Now, its stoichiometry is alpha 2 beta 2. The amino acid sequence of alpha and beta subunits is going to form the primary structure of our protein. Now, the substructures which are formed from this amino acid sequence are called the secondary structure. The subunit structure alpha and beta is called the tertiary structure, and when it comes in a stoichiometry of alpha 2 beta 2, its structure is called the quaternary structure. We are going to have further insights into this chapter, will basically give us a detailed view of these 4 levels of protein structure. Now, coming on to the primary structure of a protein, remember first in 1953. So, in 1953 it was first amino acid sequence of a protein, and the protein was bovine insulin, which was elucidated by Frederick Sanger. Bovine insulin is basically composed of 2 polypeptide chains, A and B. A comprises of 21 amino acids and B comprises of 30 amino acids, which are joined to each other either by the intra or the inter chain disulfide bonds. So, intra disulfide bonds would be the one within the A subunit or within the B subunit. When it is A linked to B via a disulfide bond, it is the interchain disulfide bond. Sequencing is generally carried out by first deciphering what is present at the N terminus. So, deciphering the N terminal amino acid of a polypeptide chain is the first step we are going to know, we want to study in the protein sequencing. This is done by labeling the amino group of amino acid at the N terminus by what is a molecule which is known as 1-fluoro 2,4-dinitrobenzene or FDNB. This is the same compound which even Frederick Sanger used for the N-terminal determination. What else can be used in dancyl chloride or dapsyl chloride? We can see in this particular figure or table, the primary structure is basically the sequence of amino acid in a polypeptide which is being stabilized by a peptide bond. Example is again which we just discussed is of a hemoglobin molecule. So the two amino acids, their sequence, whether glycine appears first or serine, then aspartate, then cysteine, this forms the sequence of amino acids forms the primary structure. Secondary structure is basically the formation of alpha helices and beta pleated sheets in our polypeptide chain, and this is stabilized by the hydrogen bonding between groups along with the peptide bonded backbone. So remember, this is the polypeptide chain and this is being stabilized by hydrogen bond, as we can see in this particular figure. Tertiary structure, the next level of protein structure, is basically the overall three-dimensional shape of a polypeptide. Now in this particular case, it could be containing a mixture of alpha helices, beta sheets or beta turns. Moving on to the last level of protein structure, which is the quaternary structure. It is the shape which is assumed by the combinations of polypeptides. So it is the subunit composition of a protein. Remember, in case of hemoglobin, it is the structure which is taken up by the alpha 2 beta 2, and this is further is going to get stabilized by the bonds and other interactions between the various side groups and between the peptide backbone of different polypeptides. Now moving again on to the N-terminal determination, remember we talked about FDNB, dancyl chloride, dapsyl chloride. So when Sanger did it initially, the N-terminal determination of bovine insulin, which we know today that contains two polypeptide chains, when he performed the N-terminal determination of this bovine insulin, he got two amino acid residues, two different amino acid residues which were phenylalanine and glycine, suggesting that it's a heterodimer. Now in this figure we can see the primary structure of bovine insulin. The chain B contains 30 amino acids, chain A contains 21 amino acids. Combinedly bovine insulin is of 51 amino acids. Now shown in this orange are the disulfide bonds. This one is a intra-chain disulfide bond because within a chain A, and these two bonds are inter-chain disulfide bonds because they are linking A chain with the B chain. Now this A chain, as we have just seen, is comprising of 21 amino acid, and we can just look at what is present at the N terminus is a glycine amino acid. And in the B chain, which is comprising of 30 amino acids, has phenylalanine at the N terminus. Before we move deeper into understanding what is the second level or the secondary level of protein structure, we need to understand certain terms. There is a term called conformation. Now what conformation means is the arrangement of atoms in a protein is called its conformation. A conformational change, these terms we'll keep encountering in the protein structure. So a conformational change is an outcome of rotation around a single bond and without breaking any covalent bonds. So the most stable conformation of a protein under given set of conditions is the conformation which is called like thermodynamically most stable, having lowest Gibbs free energy. Most stable conformations exist for a protein defined by a set of conditions such as the binding of ligand. So this was in terms of conformation, most stable conformation, and the various factors which may stabilize it. The native conformation or the state of a protein is the one which is like most stable form under given set of conditions. So if we say the native form of a protein, it means the most stable under these particular set of conditions. Stability of a protein is defined as the tendency to maintain our native state or conformation. Now, different conformations which are assumed by a protein have different degrees of conformational entropy. The unfolded state, we can like calculate, it should be having the maximum entropy because it is having maximum randomness. Disulfide bonds and multiple weak interactions such as hydrophobic interactions, ionic interactions stabilize these native conformation of a protein. Now, protein folds in such a way that the hydrophobic residues, now we have a structure of a protein, wherever the hydrophobic residues appear, they fold in such a way that takes it away from water so that it forms a hydrophobic core. So, it is embedded well inside the protein. And what is there on the exterior of the protein are basically the polar residues. Thus, the protein interior has many such hydrophobic interactions. That is why it is aptly called the hydrophobic core. The aqueous milieu around the protein forms a solvation shell or a layer around it, which is like largely defined by the hydrogen bonds that the protein is going to make with the surrounding water. Largely the proteins fold burying inside nearly all the hydrophobic interaction which is like inside the core, and what appears on the surface are the polar residues which tend to stay onto the surface. We can see this particular figure. This is shown in blue is a polypeptide backbone. This is hydrophobic interactions. This is how a hydrogen bond is formed. This is a disulfide bridge, and this is the ionic interaction which is like positive interaction between a positive and a negative. So, these are various different interactions which stabilize what is known as the native conformation or the most stable conformation of a protein under given set of conditions. Are going to now learn about what is known as a peptide bond. Peptide bond was initially worked upon by Linus Pauling and Robert Corey. They deciphered that peptide bond is planar as an outcome of that the CN bond length is shorter than that of a amine. They noticed that resonance basically existed between the carbonyl carbon and the amide nitrogen components of a peptide bond. So, basically in this CONH resonance exists. The oxygen which is like O in the CO of peptide bond and the hydrogen H in this NH of peptide bond. So, this O and H lie in transpositions. As we can see in this figure, they are lying in the transposition. There is no free rotation which can be seen around the peptide bond just because it possesses a double bond character. Hence, it is rigid and is planar. Also, the oxygen which is C of the carbonyl carbon has a partial negative charge, and the nitrogen possesses a partial positive charge. So, this forms the small electric dipole. By virtue of this positive and negative coming from oxygen and nitrogen, they are going to form a small electric dipole. The only bonds where rotation is possible, we can see in this figure, is the N-C alpha and C alpha C bonds. The bond angles upon rotation of C alpha C bond are called the psi angle, while the bond angle upon rotation of N C alpha bond is called the phi angle. Now, these phi and psi angles have to be between minus 180 degree to plus 180 degree. This is a depiction of a peptide bond. It is planar, which is shown by this shading in blue, and has a partial double bond character. We can see it is planar as well as the oxygen and the hydrogen are lying in trans to each other. Now, this partial double bond character of the peptide bond imparts rigidity to it. So, rotation is only permitted. The permitted rotations are basically N C alpha, which is the phi angle, and C alpha C, which is the psi angle. The permitted rotations around the N-C alpha bond phi angle or the C alpha C bond, which is the psi angle, were plotted graphically by Dr. G.N. Ramachandran. We can see the plot gives us the permitted rotations we can have for left-handed alpha helix, right-handed alpha helix, the parallel beta sheets or the anti-parallel beta sheets. So this region basically tells us what are the permitted rotations for the phi angle and the psi angle. So this is all about the Ramachandran plot, which gave us the information of the permissible phi and psi angles for a variety of structure, whether it is alpha helix, right-handed, left-handed, or it is beta sheets, or even for collagen triple helix. Now moving on to the next level, which is the secondary structure of a protein. Structural patterns which are made from the arrangement of amino acid residues are termed as the secondary structure of a protein. Alpha helix and beta sheets are two predominant secondary structures that occur in protein. So let's learn about alpha helix. Alpha helix exists in the hair protein or the keratin. Various conformation can be assumed for a protein by rotation around single bond and rigid peptide bonds. The simplest arrangement of a polypeptide chain is the alpha helix. The polypeptide coils around a imaginary axis with side groups which are protruding out from the helix. Now the single turn of alpha helix is around 5.4 Armstrong, which is the repeating unit for the alpha helix. This figure shows how a alpha helix is arranged. So this is coiled around an imaginary axis and the hydrogen bonds are going to stabilize this structure. The various characteristics of alpha helix is permitted phi angles and the psi angles are given as minus 45 degree to minus 50 degree, while the minus 60 is the another coordinate. Amino acid residues per helical turn are 3.6. That means it is going to take a turn after 3.6 amino acid residues. So one turn basically comprises of 3.6 amino acid residues. It can be left-handed or right-handed, but remember right-handed helices exist in proteins. So we are going to call it a alpha keratin. Now the stereoisomers which exist can be either L amino acids or the D amino acids. One of the destabilizing amino acids which destabilize the alpha helix are the highly charged ones, glutamate, lysine, arginine, proline and glycine also destabilize the alpha helix. Remember, intra-hydrogen bonding is stabilizing the alpha helix. The bond which is being formed is between the first amino acid and the fourth amino acid. Remember, the turn contains 3.6 amino acid. So if it is 3.6 amino acid, first amino acid is going to face the fourth amino acid and rest comes under this turn. So the charge of the side chains can destabilize the helix. Adjacent glutamate, arginine, lysine, which are like highly charged at neutral pH, they impede the formation of alpha helix. Similarly, proline, we know about its structure. With a ring structure, it basically introduces a kink in the helix and destabilizes it. So this was all we wanted to know about the alpha helix with its example alpha keratin, which is the hair protein. Let's move on to the other secondary structure, which is the beta sheet. Beta sheet is a zigzag extended conformation of a polypeptide. The intramolecular hydrogen bonding is formed between adjacent segments of a polypeptide. The adjacent segments can be in parallel or the anti-parallel orientation. Apart from beta sheets, beta turns are also important components of the protein structure. So let's see this beta pleated structure. This shows basically a anti-palliar orientation shown by these arrows. We can see there is hydrogen bonding again, which is stabilizing the secondary structure of a protein. Now this is one of the beta strands which is shown as a flat arrow pointing towards the carboxyl end. So this is and in the beta pleated sheet in the anti-palliar orientation. So this was with in terms of the beta pleated sheet. There are other, apart from these beta sheets, beta turns are also important components of a protein structure that basically connect the two adjacent segments of the anti-palliar beta sheet. Those are also again stabilized by hydrogen bonding between the first and the fourth amino acid. Glycine and proline appear in the beta turns. So we can see this is a type 1 and type 2 beta turn containing the glycine and proline. Now this was what we wanted to learn about the primary and the secondary structure. The third level of protein structure is the tertiary structure. The three-dimensional arrangement of all atoms in a protein is termed as the tertiary structure. When the polypeptides fold in a spherical shape, they are called as globular proteins, while the fibrous proteins are going to have the extended conformation. Quaternary structure, which is the fourth and the last level of protein structure, it is the arrangement of subunits, whether it is same or different. The subunits can be same or different in a protein forms the quaternary structure. So it can be a homodimer, trimer, or heterodimer or trimer. So we can see in this figure how our fibrous proteins look like and how our globular proteins look like. So globular protein has a more of a spherical shape. Myoglobin is shown as an example, while collagen is a fibrous protein and has a extended conformation. It is not going to come in a spherical shape. Let's learn about fibrous proteins first. Alpha keratin and collagen are two examples we can look for fibrous proteins. They have been assigned structural function. Hydrophobic amino acids are present in the interior and on the surface of fibrous proteins. Remember, in the spherical molecules or the globular protein like hemoglobin, we studied that it is going to have the hydrophobic core. All the hydrophobic amino acids are going to get embedded inside the protein, forming the core, and polar residues onto the outside. While in case of fibrous proteins, these hydrophobic amino acids can be present on the interior or onto the surface of the fibrous proteins, conferring them basically with the property of insolubility of water. This insolubility is going to come from hydrophobic residues present onto the surface. For example, alpha keratin is rich in alanine, leucine, isoleucine, valine, methionine and phenylalanine. Now these alpha keratin are found in hair, nails and make up basically the outer skin layer in us. The helix of this particular alpha keratin has right-handedness, have coil structure. Two parallel strands wrap around each other to form a coiled coil with left-handedness. Hydrophobic residues of the two helices interact to form this coiled coil structure. Here the structure is assumed by single polypeptide chain will be called its tertiary structure, while the two helices form the quaternary structure. Many such coiled coil form the structure of a protein. Let's look at this figure. This is typically alpha helix. We studied it in secondary structure. The further folding of two alpha helices is going to be called as coiled coil structure. This is a protofilament which is shown is a pair of coiled coils, while the filament is going to contain 4 right hand twisted protofibrils. Now this is how the structure of hair looks like. So beta sheets exist in the silk protein, silk fibrin. It is rich in alanine and glycine residues. extensive hydrogen bonding stabilizes its structure. So this diagram shows you how they are arranged in silk fibrion. We can see extensive hydrogen bonding, the peptide bonds linking the two chains, whether it is in parallel or anti-parallel orientation. This is how exactly this silk fibrion looks like. Moving on to, we just learnt about the fibrous proteins. Moving on to the globular proteins. Remember, globular proteins are the spherical proteins. They do not have extended conformation the way we saw for the fibrous proteins. They are going to fold into compact spherical shape. Enzymes, immunoglobulins, myoglobin are essentially few examples of globular proteins. Myoglobin is a single polypeptide of 153 amino acids. It is a oxygen binding protein of the muscle and carries also a heme group. That means heme group is basically iron plus protoporphyrin. This is a structure shown for myoglobin carrying heme. Heme is basically the one which is going to carry the oxygen to the muscles. This is equivalent to one of the chains of hemoglobin. It can be a alpha chain or a beta chain. The beta chain of hemoglobin we learnt is a single polypeptide chain with a heme group. This equates with that of the myoglobin structure. However, this is the tertiary structure of hemoglobin. If we move on to the quaternary structure of hemoglobin, so it is going to have 2 alpha subunits and 2 beta subunits. So hemoglobin, which is the oxygen carrier of blood, has a quaternary structure which is comprised of like 4 polypeptides, 2 alpha and 2 beta. Hence alpha 2, beta 2. These alpha and beta polypeptides resemble the myoglobin structure. Myoglobin comprises of heme and 8 alpha helices which are interrupted by beta turns. We can see in this figure how they are being interrupted by the beta turns. We can actually count these alpha helices, and these are the beta turns which are forming either the myoglobin structure or the one of the chains of hemoglobin. And this is how a heme group looks like. This is a iron at the center and a protoporphyrin ring. This was in terms of the structure of myoglobin and the single polypeptide of hemoglobin. We have just seen that 7 alpha helices and heme group form the structure of these oxygen binding proteins. So this is the tertiary structure, and this is the heme group which is basically containing iron should be in Fe2 positive state. Hydrophobic residues tend to stay away from water in globular proteins. That is why they are embedded inside the core of the protein, which is not true in the case of fibrous proteins. They are embedded inside the core of the protein molecule, which is called the hydrophobic core. On the surface, what we are going to have in case of globular proteins are the polar hydrophilic residues. The heme group sits in the crevice. Iron in this heme is in Fe2 positive state, which can bind to oxygen. Remember, in Fe3 positive state, it cannot bind to oxygen. To summarize what we learnt in this module, protein structure comprises of 4 levels: primary, secondary, tertiary and quaternary structure. The amino acid sequence of a polypeptide forms what is known as the primary structure of a protein, while the secondary structure has 2 predominant forms. Either it can be alpha helix or beta sheets. We also saw how beta turns look like. Tertiary structure is the spatial arrangement of atoms in space, while different polypeptides join to form the quaternary structure of the protein. We learned the distinction between tertiary and quaternary structures of protein, taking hemoglobin and myoglobin as examples.